Welcome, dear students. Today we shall talk about the next green revolution and the modern supercrops. GR2 is already beginning to take place, and it is happening in low income countries as well as emerging economies. Low income countries, many of them in sub Saharan Africa, still have very low productive agricultural systems. In these areas, chronic hunger and poverty continue to be a daunting problem, and they face the age old constraints to enhancing productivity, growth, such as the lack of technology, poor market infrastructure, inappropriate institutions, and an enabling policy environment. Emerging economies, including much of Asia, where gains from the first GR were concentrated, are well on their way to agricultural modernization and structural transformation. The challenge for agriculture now is to integrate smallholders into value chains, maintain their competitiveness, and close the urban income gap. Enhancing staple crop supplies and sustaining productivity gains continue to be important despite declining per capita cereal consumption to meet the demands of population growth and demand for feed grain. A confluence of factors has come together in recent years to generate renewed interest in agriculture and spur the early stages of green revolution. Second, in the low income countries, continued levels of food deficits and the reliance on the food aid and food imports have reintroduced agriculture as an engine of growth on the policy agenda. African leaders have acknowledged that agriculture plays a critical role in their development process, and the lack of investment in the sector would only leave them farther behind. The Comprehensive African Agriculture Development Program, that is, the agriculture program of the New Partnership for Africa's Development, an initiative of the African Union, declaration of 2006, and resulting pledges by African heads of state to increase agricultural investment showed their commitment to improve the agriculture sector. There is also an increasing awareness of the detrimental impacts of climate change on food security, especially for tropical agriculture systems in low income countries. In the emerging economies, growing private sector interest in investing in the agricultural sector has created an agricultural renaissance. Supermarkets are spreading rapidly across urban areas in emerging economies and encouraging national and multinational agribusiness investments along the fresh produce value chains in these countries. Consequently, traditional staple crop systems are diversifying into high value horticulture and livestock production. Private sector has also made significant investments in other commercial crops for fiber and biofuel. For example, private research and development and supply chains have been the primary driver behind the rapid rise of BT cotton production across Asia and Latin America. Despite these positive developments, inter-regional differences in productivity and poverty persist in many emerging economies. Rising demand for feed and biofuels and technological advances in breeding for stress tolerance could result in a revitalization of these areas. The rapid use of hybrid maize production in eastern India is a case in point. Finally, at the global level, there has been an increased tightening of food markets driven by population and income growth, as well as diversion of food grain for biofuel and livestock feed. As a consequence, the long-term declining trend in real food prices observed worldwide since 1975 leveled off by 2005. The food price crisis of 2008, sustained high prices, and more recent peaks observed in 2011 and 2012 have brought agriculture back onto global and national agendas. By 2050, global population is projected to increase by about one-third, which will require a 70% increase in food production. To meet this, Green Revolution Second must continue to focus on shifting the yield frontier for the major staples. Increasing cereal productivity not only meets demand for staples, it also allows for the release of land to diversify into high value crops and movement of labor out of agriculture, where other economic opportunities provide greater returns. Green Revolution Second must also focus on improving tolerance to stresses, both climatic and biotic. Improved varieties that are tolerant to drought or submergence enhance smallholder productivity in marginal environments and provide tools to adapt to climate change. Epidemics such as the recent UG99 wheat stem rust infestation and new virulent strain resistant to improved varieties that emerged at a time when research on rust resistance had largely stopped, underscores the necessity of continued investments to maintain resistance to pests and diseases to avoid future shocks. Finally, technologies to increase input use efficiency and improve management practices are necessary to ensure the competitiveness and sustainability of production systems. The international public goods research continues to play a critical role, but in contrast to the first Green Revolution, the context in which the CGIAR operates has changed significantly. Nurses in many emerging countries have become research leaders in their own right, which is especially true of China and Brazil. The multinational life science companies are now the leading source of innovation in agricultural science, especially biotechnology. New partnerships can channel the expertise of the private sector and advance national programs in emerging countries to benefit the low-income countries. In 2007, the CGIAR began a major reform process to better address the changing context. It is still too early to say whether the system itself will be able to reorient itself, but there are definite signs that individual centers are starting to work innovatively. For example, IRRI partnered with the Beijing Genomics Institute to carry out genetic fingerprinting of IRRI's entire gene bank collection, which will then become publicly available data. Similarly, CIMMYT is developing drought-tolerant maize for Africa through a partnership with Monsanto, which provided proprietary germplasm that CIMMYT incorporated into high-yielding maize varieties adapted to African conditions. There are also emerging examples of advanced nurses leading global public good efforts with the CGIAR as a partner and collaborator. Exemplary cases include the partnership between IRRI and China to develop photosynthesis efficient C4 rice, as well as the Global Cassava Partnership for Genetic Improvement and International Alliance of Research Institutes. The CGIAR also needs to become clearer in terms of the work on which it focuses and when it is hands off to the nurse. For instance, the CGIAR centers could hand over improved breeding materials to the nurse and leave it up to them to complete the adaptation and varietal development process. The CGIAR should also devolve the activities associated with technology diffusion to the nurse, private sector, and non-governmental organization partners. The CGIAR has had limited success in generating and diffusing technologies and practices that enhance resource and input use efficiency, thereby contributing to improved competitiveness and sustainability. The call in the work by Conway for a doubly green revolution, which is repeated in his latest book, is important for the CGIAR and the NARCIS to heed. The point that this work repeatedly makes is that understanding the underlying science is crucial to developing effective solutions. Improved understanding of tropical and subtropical agroecologies is an important global public good that contributes to innovation and new sustainable resource management practices. The emphasis of global public good research in resource management must be on such strategic knowledge generation rather than development of location-specific techniques and products. The emerging digital revolution provides new opportunities for smarter use of agricultural resources. Remote sensing and spatial mapping technologies allow for better targeting and monitoring of agricultural investments. Cell phones and other information and communication technologies can contribute to smarter applications of water, fertilizers, and other inputs. The adaptation of precision agriculture techniques for developing country smallholder agriculture conditions could have significant global public good benefits. For another green revolution to happen between now and 2050, there are two competing visions. One is high-tech, with a heavy emphasis on continuing Borlaug's work of breeding better crops, but with modern genetic techniques. The next green revolution will supercharge the tools of the old one, says Robert Fraley, Chief Technology Officer at Monsanto and a winner of the prestigious World Food Prize in 2013. Scientists, he argues, can now identify and manipulate a huge variety of plant genes for traits like disease resistance and drought tolerance. That's going to make farming more productive and resilient. Now let us talk about supercrops. Supercrops are crops that have higher yield and are crossbred to protect from pests, disease, drought, heat, and floods. In most developing countries, the majority of small-scale farmers use traditional crop varieties, which give low yields and may be vulnerable to drought, heat, disease, and other stresses. Modern improved varieties offer much higher yields, better quality, and more stable production. The International Center for Agricultural Research in the dry areas and its partners have developed improved varieties of a range of crops: wheat, barley, lentil, faba bean, chickpea, grass pea, field pea, and forage crops. The new varieties are suitable for rain-fed agriculture in areas where rainfall is low and erratic. They offer higher and more stable yields and high tolerance to resistance to diseases, insect pests, drought, heat, cold, parasitic weeds, and other stress factors. Some varieties also offer large improvements in bread making quality, nutritional value, and other traits. The signature technology of this approach, and the one that has brought both success and controversy to Monsanto, is genetically modified crops. First released in the 1990s, they have been adopted by 28 countries and planted on 11% of the world's arable land, including half of the cropland in the United States. About 90% of the corn, cotton, and soybeans grown in the United States are genetically modified. Americans have been eating genetically modified products for nearly 2 decades now. But in Europe and much of Africa, debates over the safety and environmental effects of genetically modified crops have largely blocked their use. Proponents like Fraley say such crops have prevented billions of dollars in losses in the US alone and have actually benefited the environment. A recent study by the US Department of Agriculture found that pesticide use on corn crops has dropped 90% since the introduction of Bt corn, which contains genes from the bacterium Bacillus thuringiensis that helps it ward off corn borers and other pests. Reports from China indicate that harmful aphids have decreased and ladybugs and other beneficial insects have increased in provinces where GM cotton has been planted. The particular GM crops Fraley pioneered at Monsanto have been profitable for the company, but many farmers have not helped sell the cause of high-tech agriculture to the public. Monsanto's Roundup Ready crops are genetically modified to be immune to the herbicide Roundup, which Monsanto also manufactures. That means farmers can spray the herbicides freely to eliminate weeds without damaging their GM corn, cotton, or soya beans. Their contract with Monsanto does not allow them to save seeds for planting. They must purchase its patented seeds each year. Though there is no clear evidence that Roundup or Roundup Ready crops are unsafe, proponents of an alternative vision of agriculture see those expensive GM seeds as a costly input to a broken system. Modern agriculture, they say, already relies too heavily on synthetic fertilizers and pesticides. Not only are they unaffordable for small farmers, but they pollute land, water, and air. Synthetic fertilizers are manufactured using fossil fuels and they themselves emit potent greenhouse gases when they are applied to fields. The choice is clear, says Hans Herren, another World Food Prize laureate and the director of BioVision, a Swiss non-profit. We need a farming system that is much more mindful of the landscape and ecological resources. We need to change the paradigm of the Green Revolution. Heavy input agriculture has no future. We need something different. There are ways to deter pests and increase yields, he thinks, that are more suitable for the farmers of the world. The miracle rice IR8 produced by IRRI, a non-profit, was funded by the Ford and Rockefeller Foundations in 1960. A plant pathologist named Peter Jennings began a series of crossbreeding experiments. He had 10,000 varieties of rice seeds to work with. A cross between a dwarf strain from Taiwan and a taller variety from Indonesia created the fast-growing, high-yielding strain which revolutionized rice production in Asia and was later known as India Rice 8 for its role in preventing famine in the country. When the Green Revolution began in 1960s, it was before the revolution in molecular genetics. IR8, the first miracle rice, was bred without the knowledge of the genes that blessed it with high yields. Breeders today can zero in on genes, but they still use traditional techniques and ever more complex pedigrees. That's how they have created rice varieties adapted to rising sea levels, including Swarna Sub 1, popular in India, and IR64 Sub 1. For many decades, IRRI focused on improving traditional varieties of rice grown in fields that are flooded at planting time. Lately, it has shifted its attention to climate change. It now offers drought-tolerant varieties, including one that can be planted in dry fields and subsist on rainfall as corn and wheat do. There is a salt-tolerant rice for countries like Bangladesh, where rising seas are poisoning rice fields. Only a few of the rice varieties at IRRI are GM crops in the sense that they contain a gene transferred from a different species, and none of those are publicly available yet. One is golden rice, which contains genes from the corn that allow it to produce beta carotene. Its purpose is to combat the global scourge of vitamin A deficiency. IRRI creates GM varieties only as a last resort when it can't find the desired trait in rice itself. In 2004, an international consortium of researchers mapped the entire rice genome, which comprises some 40,000 individual genes. Since then, researchers around the world have been pinpointing genes that control valuable traits and can be selected directly. In 2006, for example, plant pathologist Pamela Ronald of the University of California, Davis, isolated a gene called SAB1 from an East Indian rice variety. The East Indian rice has one remarkable characteristic. It can survive for 2 weeks under water, while as most varieties die after 3 days. The researchers at IRRI cross-pollinated SAB1 rice with a high-yielding, flavorful variety called Swarna, which is popular in India and Bangladesh. They then screened the DNA to determine which seedlings had actually inherited the SAB1 gene. The technology called marker-assisted breeding is more accurate and saves time. The researchers didn't have to plant the seedlings, grow them, and then submerge them for 2 weeks to see which would survive. The new flood-tolerant rice called Swarna Sab1 has been planted by nearly 4 million farmers in Asia, where every year floods destroy about 50 million acres of rice. The most marginal farmers reaped the most benefit. The Institute's most ambitious project would transform rice fundamentally and perhaps increase yields dramatically. Rice, wheat, and many other plants use a type of photosynthesis known as C3 for the 3-carbon compound they produce when sunlight is absorbed. Corn, sugarcane, and some other plants use C4 photosynthesis. Such crops require far less water and nitrogen than C3 crops do, and typically have 50% higher yields. The plan is to convert rice into a C4 crop by manipulating its own genes. C4 photosynthesis, unlike the submergence tolerance of Sab1 rice, is controlled by many genes, not just one, which makes it a challenging trait to introduce. By knocking out genes one by one, scientists at IRRI have systematically identifying all the genes responsible for photosynthesis in Citeria viridis, a small fast-growing C4 grass. So far, all the genes they have found are also present in C3 plants. The International C4 Rice Consortium aims to introduce into rice a high-capacity photosynthetic mechanism, the C4 pathway, to increase the yield. The C4 pathway is characterized by a complex combination of biochemical and anatomical specialization that ensures high CO2 partial pressure at rubiscocytes in the bundle sheath cells. Since its inception in 2008, there has been an exponential growth in synthetic biology and molecular tools. Golden Gate cloning and synthetic promoter systems have facilitated gene building block approaches, allowing multiple enzymes and metabolite transporters to be assembled and expressed from single gene constructs. The photosynthetic functionalization of the bundle sheath in rice remains an important step, and there has been some success overexpressing transcription factors in the cytokinin signaling network which influence chloroplast volume. The C4 rice project has rejuvenated the research interest in C4 photosynthesis. Comparative anatomical studies now point to critical features essential for the design. So far, little attention has been paid to the energetics. C4 photosynthesis has a greater ATP requirement, which is met by increased cyclic electron transport in bundle sheath cells. It is hypothesized that changes in energy status may drive this increased capacity for cyclic electron flow without the need for further modification. Although increasing vein density will ultimately be necessary for high efficiency C4 rice. If it succeeds, the same technique might help enhance the productivity of potatoes, wheat, and other C3 plants. It would be an unprecedented boon to food security. In theory, yields could jump by 50%. Now, in conclusion, we may say that developing country agriculture is faced with a growing set of challenges. First, meeting the demands of diet diversity resulting from rapid rising incomes, feeding rapidly growing urban populations, accessing technologies that are under the purview of proprietary protection, and gearing up for the projected negative consequences of climate change. Even as it absorbs the new challenges, the food policy making community continues to grapple with its traditional preoccupation of the persistence of hunger and poverty in low income countries, particularly in sub-Saharan Africa and lagging regions of the emerging economies. Harnessing the best of scientific knowledge and technological breakthroughs is crucial for Green Revolution Second as we attempt to re-establish agricultural innovation and production systems to meet today's complex challenges. New global public goods are needed that focus on shifting the yield frontier, increasing resistance to stress, and improving competitiveness and sustainability. The number of alternate suppliers of agricultural technologies, specifically seed-based technologies, has expanded rapidly over the last 2 decades. Strong nurses and the private sector have become major players in the research, generation, and release of new varieties. Even non-governmental organizations and civil society organizations are becoming active in the developing community seed systems. Innovative partnerships are needed across the entire research and development value chain to channel the varied expertise to enhancing smallholder productivity growth. At the country level, public policy can play an important role in ensuring that new innovations reach and benefit smallholders and encouraging the sustainable use of natural resources. This role requires policies that emphasize agriculture as an engine of growth and poverty reduction, enhance competitiveness of modernizing agricultural systems, and focus on sustaining the resource base by correcting distortions that create incentives for unsustainable use. Both infrastructure investments and institutional reform can help create the enabling environment for smallholder productivity growth. Furthermore, a pro-business policy environment that includes intellectual property protection, reduced trade barriers, and a transparent biosafety procedure will lead to additional private sector research investments in the emerging economies. However, the opportunities to meet these needs are not without concurrent challenges. in the areas of international coordination of public good research, weak R&D, and policy capacity among low-income developing countries, and increasing demands for immediate results. Climate change will also stress agricultural systems in poor countries as well as the capacity of the suppliers of public good research and development. Implementing a Green Revolution second will have to contend with all of these challenges and sequence innovations over time to succeed in achieving sustainable change. So, dear students, it was all about today's lecture regarding the Green Revolution second and the modern supercrops. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.